Okay, so Chapter 8, how cells release chemical energy. And the point of the last, chapter is what? We took light energy and made ATP, NADPH, and then we took that to the Calvin cycle and made what? Glucose, right? Now what we're going to do is focus on taking that glucose and breaking it down for energy. And that's the point of now. We take glucose, break it down into ATP, a usable form of energy. So where's this going to happen? This is going to happen inside plants. Now, we just talked about the other thing happening in plants, right? Why does this happen in plants? Well, photosynthesis is going to happen in leaves, where there are pigments, but plants are made up of a lot of cells that are not photosynthetic. So, for example, the root. So they're going to take that glucose and take it to other cells in their body, in their structure, that cannot do photosynthesis. We're going to take that glucose, break it down for ATP. Okay. When we do this, there's two ways we can do this, two broad ways. We can do this through aerobic respiration that requires oxygen, or anaerobic respiration that does not. But the point is this: there are different pathways to break down the products of photosynthesis. Aerobic respiration is good because it makes a lot of ATP from one glucose molecule. Downside is it needs oxygen and it's a slower process. We also have anaerobic respiration. Now this is going to make fewer ATP, but it's very fast. Okay, it's not a multi-step process. Okay. Now, sometimes anaerobic respiration is referred to loosely as fermentation. Either case, they're not either one, fermentation or anaerobic respiration. In this class, they're essentially interchangeable for the purposes of this class. Now, aerobic respiration or fermentation / anaerobic respiration both begin with the breakdown of glucose in a process called glycolysis. And it's going to take one glucose molecule. You're going to put some ATP in it. You're going to get some ATP out. End of the day, you're going to get two pyruvate molecules. Now this pyruvate molecule has a choice. It can continue to aerobic respiration or it can go to fermentation / anaerobic respiration. Okay. Now aerobic respiration starting with glucose has three major steps. One is glycolysis. Glycolysis will produce pyruvate. This happens in the cytoplasm. That pyruvate will enter into a reaction to form acetyl CoA, and that acetyl CoA will then enter into the mitochondria and undergo the Krebs cycle and electron transfer phosphorylation. Now, that's a lot there. So what are we really saying here? What are we saying is that we start with glucose. We then can break glucose down into pyruvate, and that pyruvate has two options. It goes to anaerobic respiration or aerobic respiration. But we're going to take that pyruvate into the mitochondria, break it down, and make lots and lots of ATP from one pyruvate. But what's the broad picture here? What's the big picture? We're taking glucose. It's like a $1,000 bill. And we're doing steps to make it into a stack of $20 bills, because we can use those $20 bills everywhere in the cell. And that's the greater point of what's going on here. Glucose is a carrier of energy. It has a lot of energy in it, but we can't use it. Just like a $1,000 bill has a lot of value in it, but you can't use it. Okay? Because no one takes $1,000 bills. So what are we going to do? The cell is going to take glucose and do these steps of reactions, both in the cytoplasm and in the mitochondria, and ultimately is going to come out with a stack of $20 bills. With that stack of $20 bills are multiple ATPs. It can then take those ATPs elsewhere in the cell and then use them as a source of energy that will be accepted everywhere. So that's the major point here. Okay, that's the big picture. Okay, we're taking one form of energy, glucose, and doing several conversions, enzymatic reactions to that glucose in order to make a small energy packet, ATP, that we can use everywhere to make our muscles move, to push ions against their gradients, to do all sorts of things that we use to live, okay? Our bodily processes are predicated upon the presence of ATP. How do we get ATP, this $20 bill? We process glucose, which is a $1,000 bill, to give us a stack of 20s. That is what is happening. Now, aerobic respiration is one way to do that processing. And the nice thing with aerobic respiration is a lot of ATPs can be generated from one glucose molecule. So what goes into this? You have to know what goes in. Glucose goes in, oxygen goes in, carbon dioxide comes out, and water comes out. Where is oxygen going to go in? That's going to go in the electron transfer phosphorylation at the bottom, and we're talking about this. This is just an overview. Okay, so know what goes in and what comes out. So the first stage of this is glycolysis. This happens in the cytoplasm, and it will produce two molecules of pyruvate. Okay, so we take a glucose, one glucose, 6-carbon compound, and enter into the process of glycolysis. And what happens? We have to put in 2 ATP, but we get 4 ATP as a result, so we have a 2 net increase in ATP. We also form 2 pyruvate molecules as a process, as well. So, what goes into glycolysis? Glucose. What comes out? ATP. Okay? And ultimately pyruvate. So after that pyruvate is made, we are now in the mitochondria. Before we go to the mitochondria, we have to form pyruvate into acetyl CoA. But once we've done that, we are now in the Krebs cycle, and we can take that acetyl CoA, enter into the Krebs cycle, and produce a few ATP and several hydrogen proton carriers called NADH and FADH2, which will then be converted to ATP. So what do I mean by that? The NADH and the FADH2 are proton carriers. These are going to hold protons, and these you can think of as coupons, if you will, for ATP. Okay? These NADHs and FADH2s will be used as coupons where we can take them to the next step, electron transfer phosphorylation, and exchange those for ATP. Okay? You can have 3 ATP made from 1 NADH and 2 ATP made from 1 FADH2. So think of these as coupons for ATP. These coupons are made in the Krebs cycle, but they are cashed in during electron transfer phosphorylation, and that's the third step. Okay. Third step is we're going to take those in the diagram on the right, see the 8 NADHs and 2 FADH2s? Those are coupons for ATP. We're going to take those to electron transfer phosphorylation, and we're going to have our big payday. We're going to make 30-some odd ATPs as a direct result of those NADHs and FADH2s, those coupons. We're going to exchange those coupons in for many, many, many type many, many, many ATP molecules. Now, oxygen comes into play here also because we're going to be moving around protons and we're going to be moving around electrons. But we got to get rid of those electrons somehow. Okay, we don't have a carpet we can sweep them under. They just don't go away. We've got to somehow get rid of them. How we do that is we use oxygen, and that's why we breathe oxygen in. Okay, that's going to accept the electrons and the protons as a result, and that will form water. And that's why we breathe out water. We have formed water as a result of aerobic metabolism, aerobic respiration. Okay, so we're going to start. We talked about 3 steps as an overview. Now we're going to go into detail about each of those steps. Okay, glycolysis. First step, what goes into glycolysis? Glucose. What comes out of glycolysis? Pyruvate, ATP, and NADH. Important ones here, pyruvate and ATP. Okay. A few ATP are going to come out. Not a lot, just a few. One other thing, this happens in the cytoplasm. So glycolysis happens in the cytoplasm. So to summarize, that's a good summary. At the top, we have 6 dots, 6 black dots. That's glucose. We have to put in 2 ATPs. Okay? That's going to add phosphates to each end and help us break apart that one 6-carbon molecule into 2 3-carbon molecules. We then do some reactions of those 2 3-carbon molecules and ultimately do what? We make pyruvate and we make ATP. But look at the numbers here. We put in 2 ATP. We get out 4 ATP. Okay, 2 go in, 4 come out. What is the net increase? 2 ATP. So we're making a little bit of ATP from this. Not a lot. We're making a little bit of ATP, but we're still making ATP. So we put ATP in. We then get more ATP out, and we have a net increase of ATP. And we also get pyruvate. Okay, pyruvate will be important because from pyruvate, it has 2 choices. It has aerobic respiration or anaerobic respiration / fermentation. Now, what we're going to do is now focus on the aerobic respiration from pyruvate, but be aware that it can also have anaerobic respiration. Okay. Again, this happens in the cytoplasm. Now we're going to take what happened in the cytoplasm, that pyruvate, and we're going to do aerobic respiration. This primarily happens in the mitochondria, but pyruvate alone cannot enter into the mitochondria. We have got to prepare it. We've got to have this bridge step. We've got to form acetyl CoA, and this acetyl CoA, we're gonna take off a carbon dioxide off of it. So we're going to remove a carbon, and as we remove a carbon, we also remove 2 oxygens. That's one of the carbon dioxides that we breathe out, right? Humans breathe out carbon dioxide. Why? Because as we process pyruvate, we are taking carbons off of that as we do enzymatic reactions. And as we take those carbons off, they come off as CO2, carbon dioxide. Okay. So first step is we are going to prepare pyruvate by removing a carbon, or CO2, and forming acetyl CoA. This happens in the cytoplasm. Once acetyl CoA is formed, it enters into the mitochondria and enters into the Krebs cycle, where it will combine with other molecules and form what? It will release NADH and FADH2. It will also release a few ATP and CO2. A lot of things being released here. let's kind of break it down as to what's being released. 1. FADH2 and NADH are released. Why is that important? Those are coupons for ATP. We're going to take those NADH and FADH2 coupons and take them to the next step, and we're going to convert those ATPs there. Right now, they are not ATP, but we know where to get ATP from them. That's the next step. Another thing released here is ATP itself. So this process will form a couple ATP. Not a lot, but some. And then also, look what's coming off at multiple steps here. CO2. As we process these compounds, we're removing carbon as well as oxygen. And as we take those off, what happens? Well, we release carbon dioxide as a result. And this is why you release or breathe out carbon dioxide, because you're processing ultimately what comes from glucose in the cell to make ATP. Okay? So that's why you release carbon dioxide. That's kind of the point to it. Okay? And also the FADHs and NADH2s, those will be exchanged in for ATP at the next step. And this just summarizes the process. Okay, but let's not lose sight of the big picture here, right? What are we ultimately doing? We're taking one energy carrier, glucose, and we are processing that to make ATP. We're going from a $1,000 bill to a series of $20 bills, or to a stack of $20 bills. And this is the process by which we do that. We are converting one energy form into another energy form. The energy form we are making is useful to us on a cellular level. ATP is useful on a cellular level as a currency of energy. Just like $20 bills are useful to us as a source of money in the real world. Okay? What we started with this, if we could trace back that pyruvate and we just go up from that pyruvate, we would see that what? We start with glucose. And that's a source of energy too, but it's not useful to us on a molecular cellular level. ATP is, and that's what we are doing. We're converting one form of energy into another form of energy that is useful. What we're making, the ATP, is useful to us on a cellular level. And that's the point of this entire process. Okay? Don't lose sight of that. That's the key point here. We're processing one form of energy, potential energy, glucose, into another form of potential energy, ATP. Because we cannot use glucose on a cellular level to run processes. We can use ATP on a cellular level to do processes. And that's what we are doing here. As a result, we are releasing carbon dioxide during this step. Also with the Krebs cycle, remember, we're making a few ATP, not many, but we're making a lot of coupons, a lot of things we can exchange in for several ATP in the next step. Okay, so this NADH and FADH2 are not the energy carriers we want, but we can take them to the next step and change them in. It's like at a casino. These are like, casino chips, right? They're worth something when you take them up to the counter, and that's what we're going to do the next step. Okay. The big payoff. Here's where we take those coupons, the NADHs and the FADH2s, and we make ATP from them. Okay, that is the key point here, is this is going to make us 30 some odd ATP. We're going to make a lot of ATP when we take NADH and FADH2 to this electron transfer phosphorylation. These are going to contribute electrons and hydrogen protons, or H+, and as they do this, okay, so let's start on the left here. We have NADH and FADH2. Those are going to give us protons, those H pluses, and electrons. And as they move through the proteins, what's happening is we are bringing in protons inside. And we are building up a gradient inside the mitochondria of hydrogen protons. Okay. See all the H pluses inside? Okay, think of that as a dam of water, and this is the side of the dam with all the water. Now look at that, it's almost like a mushroom. Okay, it's on the far left, it's a white sphere, and it's going through the membrane, it looks like a mushroom. Well, the hydrogen protons are going to go through this, and it will physically turn like a turbine. And when that physically turns like a turbine, what happens is it produces ATP as a result. Okay? Because there's many more H pluses on the inside than the outside. Think of a dam. You have one side of the dam with a lot of water, the other side of the dam with very little water. And as you transfer the water from the side with water to without, you spin a turbine, you make electricity. Here we're going to pass protons through that ATP synthase, which is like a turbine, looks like a mushroom here, and as that thing spins, we're going to make ATP. The only way ATP came out from this, how did we make ATP? Because, far left image here, NADH and FADH2 contributed protons and electrons. So that's why they're coupons for ATP. What they contributed could make multiple ATPs, 3 from NADH and 2 from FADH2. Okay, so what's the point here? We are making lots and lots of ATP by the contribution of NADH and FADH2. But there's a problem here. Do you see the problem? We have a buildup of electrons and H+. Think of a dam, right? What if one side of the dam has a lot of water and the other side of the dam has a lot of water? Can we make electricity in that situation? No, we can't. Because there has to be less water on one side of the dam than the other to make electricity. Same thing here. We're bringing in electrons and protons, and we just can't let those things sit around. We don't have a carpet to sweep them under. We've got to have some way of clearing them from the system after we are done with them. Okay? How do we clear these from the system? The answer to that is oxygen. Okay? Oxygen's going to come in, and it's going to grab onto those hydrogens and those electrons, and guess what it makes? Water. And that's why we have to have oxygen, because that's going to clear the system of these protons and electrons. We call oxygen in this a terminal electron acceptor. Okay? Oxygen is the terminal electron acceptor. It's the end of the road for those electrons and protons. And it helps to clear the system of those such that there's a difference, a gradient later on. Okay, so we've got to get rid of those protons and electrons after we're done with them. We can't just flush them down a drain. We can't sweep them under a carpet. We've got to somehow transfer them to something else and get rid of them. And we do that with oxygen. And that's why we need oxygen for aerobic respiration. Because that's going to be our dumping ground. That's going to be how our conduit, whatever you want to say, that's going to be our method, our mode for getting rid of those H pluses and those electrons after we have used them to make ATP. And that's why we breathe in oxygen. Okay? So here's a good summary of all of it. Stage 1, we have glucose comes in. Glucose in the process of glycolysis will make a few ATP, 2 net. It'll also make 2-pyruvate. The pyruvate comes in as acetyl CoA. Acetyl CoA enters in the Krebs cycle. The Krebs cycle makes a few ATP, but it also makes a lot of coupons for ATP. That's the NADH and FADH2. Moving down on the diagram, we see electron transfer phosphorylation. Our coupons, our NADH and FADH2, enter into that electron transfer phosphorylation. They give up their protons and electrons, and they produce a lot of ATP as a result. Because they release H+, which are protons, and electrons, we got to clear those from the system. That's where oxygen comes into play. The oxygen will combine with the H+ and the electrons, and that will make water, H2O. And it clears the system of those electrons. One last thing on this, I should have mentioned this, the Krebs cycle. What's coming off the Krebs cycle? Just to reiterate, look what's coming off the Krebs cycle. Carbon dioxide. That's why we breathe out carbon dioxide. Because, why? We are processing pyruvate, okay, to make those coupons, but as a result, we're taking off carbons, and that comes off as carbon dioxide. And that's the chapter.